research that I'm going to be presenting to you today is for my Master's of Science thesis research project here at the University of Kentucky with the Forestry and Natural Resources Department. The title of our research is The Effects of Thinning Regimes on Genetic Variation of White Oak, Quercus alba, in upland oak forests of eastern Kentucky. I wanted to begin by thanking all of my collaborators and acknowledging everybody that was fundamentally essential to this project and throughout the entire project. Without the collective expertise of all the people here, this project wouldn't have been possible, and each person was a significant contributor during different phases and throughout the entire project. I would also like to express my thankfulness to our funding from the United States Department of Agriculture, the United States Forest Service, part of the USDA, the University of Kentucky College of Agriculture, Food and Environment, and also the Forest Health Research and Education Center. Next slide. So this is just a brief presentation outline. I'm going to begin with some context, starting with our problem statement, followed by a review of some terminology, and then our specific research objectives. Then we're going to go on into the experimental design and results, and we're going to wrap up the presentation with our research findings, implementation, and a discussion of future research avenues. Next slide. So Quercus alba, white oak, is an economically important species for a couple of reasons. Specifically here in this region, it's sourced heavily for bourbon barrel production, and then throughout its entire range, it's sourced as a timber species. It's ecologically important because it's attributed to providing a lot of food and habitat for wildlife species. And as well as all other oak species, we're also seeing a decline in white oak seedling recruitment into higher canopy classes. And in order to meet these economic demands, white oak is subject to many management practices to reduce that rotation age. So due to heavy selection occurring to reduce rotation ages, potentially as forest managers, we are contributing to the oak regeneration problem and the oak bottleneck occurring. Next slide. Which led us to ask this overarching question. Ultimately, can artificial selection due to management practices and industry demands produce significant effects on the characteristics and extent of genetic diversity and natural white oak tree population? Next slide. So just a review of some basic terminology. A gene is a segment of DNA that controls a trait. And an allele is just different forms or versions of a gene. And a locus is a fixed position on a chromosome where a gene or genetic marker is. And just so that everybody's clear, a locus is the singular and loci is the plural version. So if you hear me use those terms, that's what that means. So in this figure here, we see a pair of homologous chromosomes, one maternal and one would be paternal. Allele 1, a capital A, and allele 2, a little a. And so you can see in the bottom left that you have a homozygous dominant individual with 2 big A's. Then you have your heterozygous individuals, which are a mixture of 2 different alleles in the center. And then you have another example of a homozygous individual on the far right there. Next slide. So in this example, we see 2 individuals. In the top left, we see a homozygous dominant individual, and then in the bottom left, we see a heterozygous individual with associated phenotypes. In this case, their color, and then their genotype, big A, homozygous, big A, little a, heterozygous. So even when we can't see an expressed gene, as we look at the second generation, Even when we can't see an expressed gene from observations of a phenotype, it doesn't mean that that gene isn't present. And so you can see that the heterozygous individuals maintain the diversity in wild populations, both phenotypically and genotypically, where you see in this third generation a homozygous recessive individual, but showing that blue phenotype in its coloring. Next slide. So now let's look at a population level example. So let's say that we have 3 hypothetical populations of our yellow and blue fish from the previous slide. And we can also say that these populations are in what we call Hardy-Weinberg equilibrium, or HWE. And in Hardy-Weinberg equilibrium, their genetic variation will remain constant in the absence of disturbing factors. So that in this way, you can think of Hardy-Weinberg equilibrium serving as a null hypothesis in a population genetic study. And it allows us to say if a population is out of Hardy-Weinberg equilibrium, then one or more of those disturbing factors are at play. So these disturbing factors can be anything from, Ellen, go ahead and click, please. Artificial selection, which would be seen here in the form of fishing. Go ahead and click again. Another example of that would be gene flow between populations, seen here in this example as birds maybe carrying eggs or fish with eggs in them, causing gene flow to occur between these different populations. Go ahead and click, Ellen. So sometimes these disturbing factors can also cause populations to crash over time. So here we see that fishing, artificial selection, had a preference for the yellowfish. We could say maybe people preferred eating yellowfish for some reason. Go ahead and click, but maybe the blue fish are not able to adapt as much due to their genotype. So when things like global warming or rising water changes or diseases and pathogens come, maybe the yellowfish were the only resistance, and you can see that by causing a loss of bluefish and populations not in Hardy-Weinberg equilibrium and all of these extreme disturbing factors happening, we've reduced genetic variation and caused the population to crash. Next slide. So in this study, we utilized microsatellite markers or simple sequence repeat markers to assess the genetic diversity of white oak. Microsatellites are simple sequence repeats of nucleic acids made up of nucleotides, and they're flanked by primer pairs. So you can see here in this figure, individual A is a different individual than individual B, and at this same locus, they have one individual has a different version of an allele, seen there in the yellow between the red lines, than individual B. And the way that you can tell the difference and say that this microsatellite marker is polymorphic at this locus is because there's differing segment lengths of that center repeating section. In this case would be the CTT repeating 8 times in the first individual and 7 times in the second individual. So we would say that this is a polymorphic simple sequence repeat or microsatellite. And because of its polymorphism, its multi-allelic state, then we can use them to assess populations and assess the genetic diversity of populations. Next slide. So white oak genetics and genomics, similar to other forest tree species, have traditionally not been studied too much in population genetics, and that's for a variety of reasons. So white oak, like other forest trees, has very long generation times. So in comparison to studying a plant that you can get multiple generations out of over several months, it takes years to get a generation out of a white oak tree. And in addition, they have very large, complex genomes, making them difficult to study. But what we had access to were express sequence tags, or ESTs, from the Hardwood Genomics project. And we also had access to a pre-existing thinning study where the thinnings had been implemented in the 1960s and had enough time instead of us implementing them now, currently. So with all of those ideas in mind, we asked ourselves, what would our specific research objectives be? Next slide. And our first specific research objective was to identify polymorphic microsatellite markers for Quercus alba. And then to two, determine if silvicultural thinnings significantly change the heterozygosity and genetic relatedness in Quercus alba stands, leading to a loss in population diversity in the current stands and potentially future tree generations. Next slide. So now we're going to talk specifically about objective 1, our microsatellite development objective. Next slide. So from John Carlson's lab, partnered with Hardwood Genomics, we had access to 6,995 express sequence tags associated with simple sequence repeats in the white oak genome. So we used those ESTs and filtered those EST sequences and analyzed them with Primer 3.0 to design primer pairs. And Primer 3.0 sorts these ESTs by the most likely to be polymorphic, and then attempts to design primer pairs for flanking both ends of those repeating regions. And then, so from Primer 3.0, we resulted in 84 predicted simple sequence repeat microsatellite markers. And then our next step was to evaluate the predicted primer pairs using PCR and electrophoresis on the white oak DNA. Next slide. So we tested our 84 predicted sequences on reference samples of white oak DNA. And these 3 white oak DNA samples are a subset of the entire population that we studied. And these reference samples are used to calibrate all of our future allele calls in Genemapper, and they're very important because if you can maintain these reference trees over time, they can also be used as references for future studies. Next slide. I'm going to go quickly through the lab workflow, but I do want to say that all of the protocols that we used for our lab work were previously established, tested, and accepted protocols within the community, and we didn't reinvent the wheel here. We just followed those protocols. And so we began by extracting DNA from all of the white oak leaf tissue that we collected with a D&EZ plant mini kit. Then we estimated the quality and concentration of the DNA using a spectrophotometer. Then we used PCR, or polymerase chain reaction, to amplify the allele, and we evaluated that amplification using electrophoresis. Next slide. So here you can see a preliminary 2% Agarose Gel electrophoresis image. And what you see in that first yellow box is one of our reference samples. We're just going to call it DNA 1. In the second area, that is DNA 2, a second reference sample. And in the third area, that's our negative control. We did not use any white oak DNA. We actually used water, so you can know that, you know, if nothing amplified there, then you're pretty good as far as your control goes. Back to the first yellow box, on the far right side of the yellow box, you see those bands going up and down the entire length. That is our size standard that allows us to look at and say that these bands are at a certain amount. Underneath where it says DNA 1, those are the different microsatellite primer pairs that we were testing against this DNA. And you can see that those are the tested primer pairs there against that one DNA sample. What a Agarose Gel does is it just kind of allows you to visualize what's going on, but it is not sufficient to distinguish alleles. So we had to use the next analysis to evaluate the allelic composition. Next slide. So in this slide, we see some of example Genemapper allele calls. These are two different samples. The first allele peak, where it's 168, is for one of our reference samples, and the second figure is for another reference sample. But both are looking at the same microsatellite marker 19P. You can see and say that that first figure is a homozygous individual, and that one peak indicates that there is two alleles there, but they're the same segment length, so it's the same allele. So you know that that individual is a homozygous. And then down at the second figure, we would say that this individual is heterozygous at this locus because there's two peaks at two different segment lengths. So in this way, we were able to evaluate more of the allelic composition in Genemapper. Next slide. So from these series of filters that we applied to the initial 6,995 express sequence tags, we were able to confirm that 28 of these loci were in fact polymorphic from the original set of 84. Then we took 16 of those simple sequence repeat primer pairs that were the most reproducible, consistent, and used them for further studies and evaluation. Next slide. So this figure shows an export from Genemapper calling those allele peaks that I showed you just a few slides ago. And what you can see here is a pool, which is in the yellow box, shows the 3 reference samples repeating at the top, and then just below that is the microsatellite marker that we were testing or trying to amplify. And if you look there into the red box, you see that 19T did not amplify for the first reference sample for some reason. But if you look at the second reference sample, M1146, we have an allele at 159 base pairs and another allele at 162 base pairs. So those alleles are 2 separate segment lengths. So that individual is heterozygous at that locus. And then the third reference sample, M4142, only had the peak at 168 base pairs, meaning that it's a homozygous individual, and those 2 alleles were the same segment length, creating 1 peak. So in that way, you can say that this microsatellite marker is polymorphic because there's multiple versions of that allele existing. There's multiple segment lengths of that allele, and that's how we can assess how many alleles are present at these locus or loci. Next slide. So in conclusion of the first objective, we identifying microsatellite markers for assessing the diversity of Quercus alba stands in populations. We were able to identify and validate 16 polymorphic microsatellite markers and add that to the currently limited number of microsatellites available for white oak. Next slide. So moving on to objective 2, determine if silvicultural thinning significantly changed the number of heterozygous individuals and genetic relatedness in white oak stands. So we were able to have access, thankfully, to a thinning study started on the Daniel Boone National Forest by the USDA Forest Service in 1961 and 1962. And the objectives of their thinning study were to assess the effects of stand density on wood production, growth of residual trees and the quality of the residual trees left. Next slide. So when they were thinning these stands in 1961 and 1962, their selection process follows this criteria. They cut all wolf trees, and those over 9 inches in DBH. They cut trees in these half-acre plots as to evenly space the trees. They cut any other undesirable species that were not white oak, and they cut trees of any poor or undesirable qualities as far as wood and timber production go. So following this artificial selection protocol, how would artificial selection affect the genetic diversity of these white oak plots? Next slide. This is a map showing that the Danube National Forest is part there in the southeast region of the United States. It is close to McKee, Kentucky, and it is on the Danube National Forest. It's considered an upland oak forest type and part of the northern Cumberland Plateau region. And the figure on the far left just shows how the plots were arranged across this watershed. And the plots in yellow are our control plots, meaning that no thinning has occurred and they did not get thinned at the same time as the other stands. The orange plots are a medium thinning intensity, and the red plots are what we deem a high thinning intensity. Next slide. So this table just shows more how we delineated the thinning intensity labels. So you can see that the medium thinning intensities were only thinned once, the initial thinning in 1961, and then there are 2 different target basal areas for post-thinning. And you can see that the high intensity plots were thinned more than once twice, and the other 2 3 times there in the number of thinnings. So this was just a chart to show you how it broke down those different thinning intensities for our analysis. Next slide. So all of the samples that we collected from this population were foliage leaf samples. In total, we collected 225 foliage samples, and that was collected with hand, pole pruner, and in most cases firearms. Since the trees were so tall, we couldn't reach the foliage samples. And we did sample all mature white oak on our study plots, and all that means as far as mature goes for this study is that the trees were on the plot after the thinning and have survived to the sampling year when we sampled these foliage samples. These are just a couple of pictures I wanted to share with you guys from like the field and troubleshooting some of our sampling processes. Next slide. So in addition to the foliage samples, we also did GPS locations of the trees. A large thank you to Dr. Lodka and Dr. Contreras. The exact location of each tree was determined using the plot corners and then using a digital compass and laser rangefinder from those plot corners. Next slide. So for the DNA extractions and visualization of the DNA from all 225 samples, we just followed the same lab protocols that I mentioned previously. So all the DNA extractions were done with a QIAGEN plant mini kit and just followed the same protocols as we used for the 3 reference samples mentioned in Objective 1. And all of the DNA quality and concentration was visualized with a spectrophotometer. Next slide. So we took these 16 polymorphic microsatellite primer pairs we identified from objective 1 and used the same polymerase chain reaction protocol, just now only with 16 SSRs over 225 samples instead of testing all 84 across the 3 like we did in objective 1. And then we did electrophoresis on a capillary electrophoresis machine. Then this just kind of shows, next slide, this figure shows the workflow for our allele calls from the reference samples that we then use to calibrate the rest of the allele calls and then manually check. So you can see there in the top left, the X is representing our initial reference samples, which would be a subsample of all 225 samples shown in as the blue squares, ran them in a capillary electrophoresis machine, exported those capillary data files to Genemapper, and used our allele calls from those reference samples in a FastProc to calibrate the rest of the allele calls for us. And then we just have to go back through and manually track those allele calls and complete this process until we have all of our allele calls for all the populations across all microsatellite markers that we tested. Next slide. So as I mentioned, we used a FastProc in combination with Genemapper to get that allelic data, and then that data was then exported and imported into GenLX, which is just an add-in to Excel, and we use that for the population genetic statistical evaluation. And then we exported some of the data that we got from that analysis into R and RStudio for evaluating the genetic relatedness data. So now I'm just going to go into some more of our specific results. So next slide, Ellen. This first figure shows the thinning effect on heterozygosity, just looking at control versus all of the thin plots, not delineating by the specific thinning treatment. So you can see here the red arrows that 3 locus for the control were out of Hardy-Weinberg equilibrium and showed a significant reduction in the expected heterozygosity to what was actually the observed heterozygosity in the population. And then if you go down to managed, there are actually 8 locus out of Hardy-Weinberg equilibrium compared to the 3 in the controlled. So by looking at this, we were like, okay, there is a significant reduction in heterozygosity between our unthinned stands and our thin stands. So then that led us to break it down a little bit more. Next slide. So then we asked, is there a difference in the residual basal area? So the basal area or the target basal area that they thinned to at the time of the thinning. So the top group with the yellow arrows shows a residual basal area thinned to 45 feet squared per acre, and the bottom is 75 feet squared per acre. And you can see that 5 were out of Hardy-Weinberg equilibrium significantly in the 45 feet squared per acre, and also the same 5 were out of Hardy-Weinberg equilibrium, the 75 feet squared per acre. So this means that there was a reduction in heterozygosity, but not a significantly different one between these 2 groups. It just seems that there is a reduction as long as there's a thinning happening. Next slide. So then we wanted to look at the number of thinnings and see if that affects the number of loci that are out of equilibrium and experience a significant reduction in heterozygosity. So the yellow arrows show 5 locus or loci out of Hardy-Weinberg equilibrium, and 1 thinning only had 3, so there is somewhat of a difference in the number of thinnings as far as the severity of the reduction of heterozygosity in the population. And then finally, we combined the number of thinnings with the residual basal area into those thinning intensity delegations that I showed you earlier in the chart. And what you see here is 5 loci out of Hardy-Weinberg equilibrium for high selection thinning intensities. Then you see 6 out of Hardy-Weinberg equilibrium for the medium, and then 3 out of Hardy-Weinberg significantly with a reduction of heterozygosity in the control. So there is a difference between the unthinned stands and the thinning, but not so much between the actual thinning regime, other than that it was thinned. So now we're going to move on to some of the genetic relatedness results that we got from the study. Next slide. These box plots show a general trend for the individuals in control plots shown as CC. So let me just tell you guys really quick, CC, CM, and MM down on the x-axis, that's just referring to the pairs of these individual trees that we were comparing. So we were looking at the mean genetic relatedness, or LRM, between each pair of individuals in the population. So CC means that the pairs were from control plots. CM means that one individual could have been in a control plot and a managed plot, the other individual, and then the third, MM, shows that both individuals from the pair were in managed plots. So this box plot just shows a general trend for individuals in the control plots to be more genetically related compared to pairs across treatments or within managed plots only. Next slide. So moving the two key multiple comparison means, this figure shows that the LRM, or genetic relatedness, is significantly lower between pairs from the managed than control, because the MMCC line is less than 0 at the 95% confidence interval. Next slide. So this figure shows the main effects of a two-way interaction in mean LRM, or genetic relatedness, of pairs from the same plot or two different plots, CC, MM, CM. Each pair's mean measure of genetic relatedness is shown on the y-axis, and the treatment type is on the x-axis. So we can see here, so if you look down at the, let's see, bottom right figure, we can see that pairs from the same plot have a higher amount of genetic relatedness than pairs that are from different plots, regardless of the treatment that is happening, thinning or no thinning. And then if we look at this top left figure, we can see that the managed plots have less genetic relatedness, or tend to, regardless if they're from the same plot or just. So this model comparison shows the general trends that both type and treatment do affect the genetic relatedness in these plots. Next slide. So then we wanted to look at health relatedness distance, and we have
I think that we are
here.
Rachel, I think that we're losing you. Continue talking, and if you cut out again, we can reset in just a few minutes. But why don't you keep going, and we'll see if this issue resolves itself.
Okay, it just started raining where I am, so I suspect that that has something to do with it. So I'll just continue and see how it goes. So what we see,
hey Rachel,
here
can
in
you
this
go back
figure,
one slide to the first slide of the model regressions there?
Okay, slide 39.
Sounds great. Slide, how about slide 40?
Okay. Okay, so this figure is just showing the trend of genetic relatedness over distance for all of the managed plots. So you calculating the Euclidean distance between the individuals in each pair that we used in this analysis. So we were able to show this in this scatter graph, the general decline as distance increases, downward trend. So now we're just going to go through the same figure, but for each plot and I'm going to say it's associated thinning intensity. So next slide. This is plot 3. It was delineated as having a medium selection intensity. Just see a general downward trend here. The next slide is plot M, medium selection intensity, and you just see a general downward trend of genetic relatedness as distance increases. Next slide. This is plot M4, a high thinning intensity plot. The steepness is not quite as dramatic here, but if you remember back to the plot design table, this plot actually had the highest trees per acre, so it's kind of an outlier, I would consider it in our data. Next slide. This is plot M11, also a high thinning intensity, and you do see a steeper general trend here. And then we see plot 10 on the next slide, and it was a high thinning intensity and showed us the steepest significant general downward trend as genetic relatedness decreases as distance increases. Next slide. So just as in summary and conclusion, between managed and thin plots compared to the control plots, the managed plots had more loci deficient in heterozygosity, and this is linked to a reduction in genetic diversity. Also, between managed thin plots compared to control plots, we have lower degrees of genetic relatedness among the trees, and we saw that the relatedness varied and decreased generally, and in some cases significantly, by distance. Next slide. So just to kind of bring everything full circle, we saw a reduction in heterozygosity in this population over these 7 plots that we studied. Typically, heterozygosity can be caused by large levels of inbreeding, which would be typical of low population numbers. Reduced heterozygosity is also associated with isolated populations, maybe ones that aren't able to get that pollen flow or gene flow between stands, and maybe just like being geographically actually isolated. But what we really believe is going on here is a reduction in heterozygosity due to selection pressures. And I wanted to talk a little bit about the Wayland effect, which is caused by, it's a reduction of heterozygosity caused by a subpopulation structure. So my question is that potentially, or can we say that these thinning regimes are creating a subpopulation structure where we have thinned and unthinned trees creating these 2 subpopulations within a population, effectively reducing heterozygosity. But what we can for sure say and attribute to a reduction in heterozygosity is that there is going to be a general decline of fitness and genetic diversity over time. With a reduction in heterozygosity, you can expect to see the loss of rare alleles in future generations, and maybe by reducing the genetic variation and genetic diversity in these populations through our thinning regimes, we're potentially contributing to the oak bottleneck issue, where these seedlings we're reducing their fitness and their adaptability, and they aren't able to compete against other species like maple and beech. Next slide. So for the genetic relatedness section, we did see a difference between the control and managed, just that the control were more genetically related generally than the managed plots. So you could say that maybe this is combating inbreeding, that thinning can combat inbreeding, but I would argue that by simply reducing the population numbers by thinning, you're kind of contributing to inbreeding, and that merits more research and more sampling in the future to further diagnose that question. Next slide, which really leads me to talk about the future research or the things that I get excited about seeing or potentially being a part of in the future. So one way that I would really like to see this project expanded is instead of sampling, what we pretty much did was an even age class. All the trees were released at the same time in 1961. It would be really interesting to study multiple age classes, so potentially sampling the seedlings in this population as well. We had 2 control plots that we consider to be natural stands or unthinned stands, and I think it would be really important to get more control plots just so that the experiment is a little bit more balanced. And then in addition, adding other populations other than the population that we looked at in that McKee watershed, or Elijah Branch, as some of the Forest Service people call it. They have another location called the Bald Rock Study Site, also on the Daniel Boone National Forest, where they did these same thinning treatments and thinning plots. So it'd be really interesting to get a multiple population comparison in the same region. And what is really exciting is maybe doing a landscape genomic and genetic study across the entire range of white oak, where you have populations that you're sampling throughout the entire range just to get a better picture of the collective adaptive genetic diversity of the species, and maybe how different management practices in different regions to meet different demands are affecting it differently across this entire range. Next slide. So just in review and just to highlight everything, our 3 main findings or our highlights is that we were able to develop 16 highly polymorphic satellites or SSR markers for white oak, for Quercus alba. We were also able to show that there is a significant reduction in heterozygosity between our control or natural stands and our thin stands, and that there is also a general trend and significant reduction in genetic relatedness on the managed plots compared to the control. Next slide. It's just my literature cited. Next slide. And at this time, I'd like to open it up for any questions.